All right, all you. Thank you very much. And before I get going, I just want to give a hearty congratulations and thanks to David for organizing this seminar all year long and getting speakers from outside, speakers from inside, and accommodating this extra May I series. So it's a lot of work and you do it very well.
Thank you all, and I'll be bothering you for next semester.
That's right. Yes. Okay, so thank you for coming. I'm going to spend today recounting a little bit of my track in learning all of this stuff. So I fair warning, last about Christmas or so, 6 months ago, I knew how to spell neural network and I knew nothing else about a neural network. And I picked up a book and I started kind of reading through it, and I got very intrigued by the possibilities that it might offer us in analyzing complex data. I started kind of working through it and stumbling around, and then I got a few folks on our in my research group involved. And so what I'm going to talk to you about today is a little bit about what a neural network is. I think some of you may not know exactly how it works, so hopefully I can enlighten that a little bit, but then I'm going to go forward in some of the applications, the first applications that I've tried to use neural networks in deep learning in, and in particular in thinking about how to model complex genetic and environmental data. So as I said, I'll talk a little bit about what a neural network is, a little simulation I did to make sure that I think it works. And then a little bit about PRS, some applications to colorectal cancer, and then circling back and kind of applying it into a colorectal cancer data set. And I'm going to end with a little bit about where I'm most interested in taking this, which is kind of adding in biological knowledge into this kind of deep learning framework to go beyond just standard kinds of deep learning approaches. So here's a simple neural network, and I'm going to use this just to try to give a sense for how these things actually work. So how many people have actually tried running a neural network analysis or taken a neural network class? Okay, a few, right? So good. So neural networks are basically set up like this. You have what's called an input layer, and that's real data. That's data that you're feeding in. So given that we do a lot of this in biostatistics, we think about inputs and outputs. So I'm going to call the inputs X. That's our set of variables that we want to use as predictors. The output on the other end is some outcome Y that we're interested in predicting or modeling. And then there's these things in the middle that are called hidden nodes. And I've drawn a very simple picture. You'll see some more complicated pictures as I go through. And a characteristic of the standard feed-forward neural network is that all nodes are basically connected. So you'll notice that each of the input nodes is connected to each of the hidden nodes, and each of those is in turn connected to the output node. And then there's these extra things at the top that I've added in, which are also connected to the downstream nodes. And those in the machine learning neural network realm are called bias nodes, which seems like an unfortunate choice of words, but I like to think of it as just like an intercept term, right? It's kind of an offset, much like an intercept is an offset in a logistic or a linear regression model. So just think of
what distinguishes the bias nodes from the other.
So the input layer X is going to be data. That's going to be your covariates or whatever that you're feeding in.
Okay.
The output is the output, and then the hidden layers are just internal nodes. You don't know what they are. That's where the magic happens. But then part of the model for what happens in the hidden nodes is these bias nodes. So just think of it as an intercept. So think of it as an X vector of all ones, right? Which is sometimes how we code an input X matrix for a linear logistic or what have you type regression.
Wouldn't that be an input?
They try they usually distinguish like real data that goes in the input node versus what's needed to just parameterize the model correctly.
Okay.
So now I've added in here a bunch of parameters, and these are called weights. And they're indexed typically by what layer and which node they're attached to. But again, you can see all nodes are basically connected. And I'm using W's for the first layer node weights, and then V for the output layer. And I've also added in just to kind of make this as clear as possible, that what you're doing is you're generating a prediction, which I'm going to call P, for whatever the target that output is Y. So I'm going to distinguish the data Y from the prediction. Think of P as Y hat, if you want to think of it in regression terms. So how does this thing actually work? So the basic goal of the whole thing is to estimate these weights, because once you estimate the weights, then you have this link between your input vector or matrix and your output Y. So it's identical to what we do in standard regression, right? We have X data, we have Y data, and we want to estimate weights, which are our betas typically. The way this works is you start with some arbitrary value of the weights, and you compute essentially the value of H1 for a given set of inputs as just a linear combination of the bias node, the W01 up at the top. So that's up here, or that feeds into H1 node. And then W11 times X plus W21 times X. So it's exactly just a linear combination of the inputs. And then you apply what's called an activation function. So that's the A out here. So it's a function of that linear predictor. Same thing for H2. So you're just taking the corresponding bias weight and the bias weight time X2, et cetera. And then that gives you then values for the hidden layer nodes H1 and H2 for a given input, right? So for each individual person in your data set, you're gonna be running this through and computing an H1 and an H2. Once you have H1 and H2, you can then feed that forward, and it's a linear combination using the V weights here to generate your final prediction. That also then is applied an activation function, right? So, and the activation function doesn't have to be the same from layer to layer. So I'm going to talk about that in just a second here. So the basic idea with the neural network is it's kind of based on the brain, that when the brain gets stimulated in whatever way, neurons fire. They either fire or they don't fire, and that causes things to happen, right? Whatever the stimulus is. So the basic idea is for the hidden nodes, we want the concept of the node fires or it doesn't fire. So that would be the equivalent here of having a step function, which at some point in the value of that linear combination, right, the W's times the X's, if it exceeds a certain value, for example, then you would say, okay, the node fires, and if it's less than that value, it doesn't fire. And so typically, if you were programming it that way, you might say that H1 is equal to 1 if that linear combination of the weights is greater than 1, or actually probably should be greater than 0, the way I drew it, and 0 elsewhere. So you have some threshold. If it's above that threshold, it fires. If it's below that threshold, it doesn't fire. So that's the concept of what we want the neural network to do. The problem with that is when we get into kind of the calculus of how it actually estimates the weights, you really want a smooth differentiable function, and this one is not. So there's a lot of alternative kinds of activation functions that are used. One very commonly used one is called the rectified linear unit, with a ReLU. And that one has a similar kind of characteristic. It's continuous. It's 0 if you're below some threshold. So maybe 0 if you're below 0. And then it's set equal to whatever that value of the linear predictor is if you're greater than 0. Okay. Seems a little unintuitive why that works, but it seems to work really well. There's modified versions of the ReLU. You can use other kinds of things. Then when you get to the output layer, things usually change a little bit, so that if your output is, your Y is continuous, then you may not apply any activation function. You just take the linear predictor as is. Think of that as kind of a linear regression style function on that last layer. If it's binary, then typically something like a sigmoid function, which is, we all know, is an x-bit function, kind of a anti-logit is used, but there is hyperbolic tangent is sometimes used, and there's a few others, but basically trying to get at estimating a probability or a prevalence. All right, any questions about that? Okay, so you feed this thing forward, you compute values of all this, you get a value of P, and then you want to find out how good it is, right? And if you start kind of in an arbitrary set of weights, probably not going to be a very good prediction. So we need some kind of a loss function, and here standard kinds of loss functions that we're familiar with are used. So if Y is continuous, typically something like the mean squared error, just the sum of the P's minus Y squareds. If it's binary, then the cross entropy function is typically used. Right? That gives us a sense then for a particular set of weights, how close are we to the truth? And based on that, then we want to make some adjustments. So this gets to what happens when we actually fit the model. So we essentially start with some set of weights, we feed it forward, compute the loss, and then we have a back propagation step. And this is where the weights are all going to get kind of jiggled and nudged in certain directions. And the directions they get nudged in are going to depend on how big the loss is and how big the difference is as a function of the change in weights as you kind of back propagate through the network. And then you repeat steps 2 and 3 until you get to some point where you think that the thing might have converged. And so this is a standard kind of machine learning situation where you've got typically a training data set, testing data set, and in the training data set, you're trying to optimize over some parameters. You might be doing some cross-validation to try to tune, and there's a number of things you can think about tuning for these things. But you get to some point where you think, that's the best model, and then you see how it works in testing data set. The back propagation is actually involves, a little bit of calculus here, kind of interesting, but it's actually really simple. So if you think about the loss function, then ultimately being this P minus Y squared, let's take that as the loss. And for the moment, let's just define Z to be the linear predictor part for this last layer. So we'll just think about what happens in the last layer weights. So P is just the activation function applied to that Z. Then we want to look at the gradient of the loss function, say as a partial derivative of L with respect to one of the weights. So I'll show you what that form looks like. We'll have the same kind of form for other weights. And it's just a simple application of the chain rule. So that breaks down into the partial of L with respect to our activation function, the activation function with respect to Z, the linear predictor, and then the derivative of Z with respect to whichever weight we're interested in looking at a derivative. So when you put those three together, it has something that looks like this. The first part, whatever the first derivative for the activation function, and then the derivative of that last little bit, the Z with respect to V1 is just going to be whatever the value of H1 is, okay? And then you do that for the other hidden nodes. This is the function of H1, so you're going to propagate that back. So you're going to have a corresponding set of partial derivatives as you work back through the function. And so when you get to the end and you go backwards and compute a loss, and then you want to try to nudge these weights, and you're computing all these little bits, and then that gives you what you use to update the set of weights for your next round. Now, you might think you want to just run all n subjects through this and turn around and go back and compute the derivatives, et cetera. But what happens in practice is that this is run in batches. So you randomly kind of reorder the X input layer, and you might take a batch of, say, only 16 subjects, feed those forward, get the loss, do the back propagation step, update the weights, and then go to the next 16, and then go to the next 16, et cetera. So you're doing this kind of in chunks. And the benefit is theoretically still gets there, and it gets there a lot more quickly than if you try to do it on all n subjects each time you go through. So that's one of the tuning parameters is the batch size. So very simple little example that I kind of started trying to learn how to do this, and sat down and programmed it in C++ so I could figure out all these steps and make sure I knew what the heck was going on. So the XOR function, which is just 4 lines of data, y is 0 if the x1 and x2 are equal to each other, and it's 1 if not. So it's x1 or x2. And if you run that through and see what happens is something that looks like this. So this is the weights that I got. Doesn't really matter what they are. They're not interpretable. We don't really care about them. What we care about is at the end of the day, is the prediction any good? And so when you run this through, you get predictions of like, 99%, et cetera. And so if you were doing any kind of classification, obviously you would say, hey, I'm going to set it to a 1 if it's above some threshold. 99 is pretty close to 1. So you'd probably call that 1, and the others you'd call it 0. This is the basic thing that, is done to say, is the picture a dog or a cat? You get a probability of a dog or a cat at the end of the day. And if it's high for a dog, you assign it to be a dog. If it's low for a dog, you assign it to be a cat, right?
I have a question related to things that are been split processing. Does the batch approach do anything to help prevent overfitting? Like if you've got a weird example, if you've got a weird sample, it only comes up once out of every 10 batches or something, or is it purely just a computational?
Yeah, that's a good question. I don't think the batching necessarily is designed to help with overfitting, but you can, there is a tuning parameter in there called dropout, which you can drop out a certain portion of your samples on every pass through the complete data set, right? Still do the batching within, and that is designed to prevent overfitting or to minimize overfitting. Yeah, so you can drop out 10% of the sample and go through, do the whole deal, but drop a different 10%, et cetera. Yeah. So I ran this for a couple different things. So the XOR function is on the left, the X and function. So that's only a 1 for Y if both are a 1. You can see what the outputs look like there. Very easily finds the 1. And then I did an additive one, and it very nicely kind of replicates what Y is. So it's totally simple, but obviously very flexible. You feed in whatever you want, hit go, and you get something that looks pretty good. That's a neural network. So what's deep learning? Deep learning is a neural network with 2 or more hidden layers. Woo, right? That's all it is. So this is an example of deep learning. And this is a famous example for image recognition where there's a nice data set out there that is hand-drawn numbers. There's 784 of them, 0 through 9 digits. And the goal is, can you identify what the number is, right? So this is obviously a 9. We all know it's a 9. Across the 784, there's different people drawing different kinds of numbers. So some of them look more like what they are. But the basic idea here is that the first layer might be picking off, is there a little curve at the top or a little curve this way or a straight line? And so the different little colors up there are meant to signify characteristics of a number that first hidden layer might be picking up and firing on if it sees a particular kind of a loop or a straight line. And then the next one is starting to piece those together and fire if you have a collection, that are in the direction to give you a loop on the top and a straight line. And then at the end of the day, you hope that thing gives you a 9, right? If you fed in a 3, different things would fire in the middle layer. So that's kind of the basic heuristic of what's going on and why you might want multiple layers, because each layer might be piecing apart a different piece of the puzzle. So I decided to go beyond the 4-element XOR function kind of thing and try this out on some simulated data that's a little more like what we see. So I simulated 10,000 subjects. I split them randomly into a training and testing data set. Continuous outcome. I generated 2 grouping variables. I'm going to just call them sex and genotype. They were just binary variables, but just so I can label them. And then I generated 11 predictor variables, 5 of which were binary, 6 were normally distributed. And then I generated a Y that depended on just a function of the X variables. Okay. And I did 2 different scenarios. So the first scenario, I generated it so that I have my 2 grouping variables up there, male and genotype. Genotype just yes, no, carrier, non-carrier. And if you look over here, I essentially generated Y with a mean that was different for each of those 4 groups. So the betas that I set up were ranged from 0.1 to 1 in this group. They were the reverse direction in this group, negative values over here and nothing going on out here. So this is what the kind of the linear predictor looks like for generating a simulated Y. That was the first simulation. And I had some squared and cubic terms down here, which I set to 0 for the first scenario. Second scenario, I did the same thing up here, but I added in a quadratic function going up for one group, quadratic down for the other, kind of a S-shaped cubic for this group and a negative S-shaped cubic for this group. So I tried to throw in a couple nonlinear terms. So what did I do? Well, I did standard linear regression, just fed in the main effects. That was a baseline model. Second one, I thought, well, you might recognize that there's some nonlinearity in the X11 term, so you might throw a squared or cubic term in. And then number 3 is the generating model, which takes 55 terms to get it right, all possible interactions. And, you know, maybe a model you may never get to if you're just sitting there doing, you know, an afternoon's worth of linear regression.
So remind me, what's the true model?
The true model is basically has different values of the parameters. So it may be easiest to look at here. So there's essentially 10 terms in each model for the expectation of Y that depend on different values of the betas for each of the inputs. So a very nasty, you know, interaction model. Some negative trends, some positive trends, some with nothing going on, etc. All right, so then what is the neural network going to look like? Well, the inputs here are relatively simple. It's the 2 binary variables and the 11 predictor variables, some amount of hidden layers, and then the output is Y. So I set this up, there's 13 input nodes. I looked at various models from 1 to 3 layers with varying numbers of nodes, ran this thing kind of 200 cycles, which means it'll cycle through the weight updating 200 times, set the batch size to 60 and the dropout 1%, which tries to prevent overfitting. The loss is a mean squared error. The activation functions, I use this ReLU for the hidden layers and just a linear for the output. And I was tinkering around with the R package Keras, which I got from Josh and a little help from John. Thank you. And then at some point, Ming-Ji gave his talk about PyTorch, and I asked him to try it out in PyTorch. So for the first scenario, which is the easier one that doesn't have the quadratic and cubic terms, if I just fit the standard linear regression model, the AUC for the training and testing data set is around 45%, 45% for the testing. If I add in the quadratic and cubic terms, it goes up. And if I fit the true model, which should be the best we can do, it's about 89%. right? Again, I don't think you may ever land on this model unless you did a fair amount of model snooping. Neural nets, I was able to recover that basically relatively simply, and it was with a 2-layer model, 7 nodes for each layer. I remember I didn't have to do anything. I fed in sex, genotype, and 11 variables. So I said, go, right? So not much human intervention. So that was kind of fun to see. For the more complicated scenario where I had a cubic thing in there, again, the model is a little different. The truth, what I think is probably the target AUC would be 81%. And I tried a bunch of different architectures. So 1 layer with this many nodes, or 2 layers, or up to 3 layers. And you can see that the AUC that you get can vary quite a bit, right? And the degree to which, you know, it looks like maybe there's some overfitting when you see a higher AUC for the training versus the testing. And so I spent a while tinkering with this and just trying different things and jotting these down on my piece of paper. And then I asked Ming Xi to run it, and he basically did it in probably about 30 minutes. And came back with a nicely tuned model and got basically better at anything that I was able to get. So still a little lower than this, and that, you know, there's a possibility that additional tuning or different architectures might do better. But again, you know, a relatively straightforward set of inputs and let the thing run. So any questions?
So for the result, you only like change the model structure, but now keep all the other hyperparameters constant and fixed, and like the batch size or dropout rate.
Yeah, I had the batch size, dropout rate. I don't actually know, maybe Ming Xi can remember, it was a while ago. Could have been differences between what I was doing and what he ended up doing. And I think he optimized a little better than I did over these different tuning parameters. But yeah, it's still relatively straightforward.
Okay. Yeah, I mean, the overfitting can be relieved if we separate the training to a real training and a validation set.
Yeah.
Between those other parameters.
Yes. Yeah. And so Ming Xi actually did a better job at that. So he did a proper cross-validation in the training step and tried to tune over these some of these hyperparameters.
I have a small question. Did you do much feature engineering in the neural net? Did you include like X squared as an input, or you just had X?
Just X. Yeah. So yeah, really it was sex, the G, and 11 X's. And I didn't put any squared or cubic terms in there. Because, you know, ostensibly neural nets are supposed to be good at finding nonlinear patterns, right? And recreating nonlinear functions. So I wanted to see if it could find it on its own, basically. If I had thrown an X squared and X11 squared and cubed in there, you know, it might have been better, and it might have, you know, more easily recovered the true model, I'm guessing. So, yeah.
I have a very general question that might not be answerable, but is there any relationship between the degree to which a neural net will overfit the data and the size of the neural net? So, for example, do they tend to overfit more when they're more complicated? Yes,
in my experience, at least, yes. And when I went through this, oops, what did I do? When I went through kind of this, you know, trying different models. I don't know if you can see it here, but the more complicated it got, I got the sense that overfitting tended to be more of a problem. So, and there's ways to try to monitor this. You can kind of graph the mean squared error as you go across these epochs, and you'll see patterns where the mean squared error for the training data set always declines, you know, because it's getting better. You can monitor the mean squared error for the testing data set, and typically that will kind of bottom out, and then at some point it'll start going up, getting worse. Right? So you can kind of keep a visual eye on this as the model is being trained, and that place where it starts to get worse is, you know, probably where you want to be. And the algorithm, at least that Ming-Chi is running, automatically picks that at best point. So it's a function of the cross validation.
But then
yes.
presumably you're then subject to winner's curse or something, right? So you probably need a third data set to really test.
No, that's right. So when Ming-Chi did this, he properly, within the 80% that I call training, he allocated a fraction of that for cross validation, and tuning and all that, and then he held out the 20% as a final step for the evaluation. So, all right.
So can you look at these weights and say what they, or what they're actually doing?
Can you look at the weights and say what they're actually doing? No, I don't think so. I mean, I wasn't even able to really figure out why it set the thing up for the simple XOR function. I mean, you can stare at this and you can see why it works. And, I wrote it down on a piece of paper, and yes, sure enough, these all work out and they give you these probabilities at the end. But when you get to anything complicated, especially a multi-layer thing, I think the weights are completely unintelligible. I think there are ways, I think there is software out there to try to look at the patterns and the weights and to try to abstract features. I don't know, you're shaking your head, so.
Yeah, there's a whole field of interpretable AI or stuff like that. I've read a few of those papers, but you can kind of like, for any given observation, you can trace the weights and the activation function backwards and sort of say like, these are the features that this node is taking its cues from.
Yeah, I think that, yeah, what's causing that node to fire, right?
Yeah.
Basically, yeah. All right, so, quick summary of the simulations. Obviously, linear regression would be nice. You can predict, you can do inference. I think if you have, very complicated models, it can be difficult. Neural nets, very good for complicated models, good for prediction. Weight's not that interpretable. But I think, we saw Juan Pablo give a nice talk last week. Downstream inference for these machine learning approaches may be possible using something like targeted learning, but it requires that kind of framework. I think this gave me the sense that, one possible use of neural networks when you're just trying to explore data versus, standard linear regression could be try both. And if you get a dramatically better prediction with a neural network, then you probably know there's something going on in there that's worth exploring. And even if you want to resort to linear regression, you may not want to settle on just a standard main effects model. All right, so I'm going to switch to polygenic risk scores. So we've seen others talk about PRS. I'm not going to belabor it too much. Basically, we have a first data set that identifies a bunch of SNPs, potentially, maybe GWAS significant. A second data resource that is used to fit a model for disease or outcome on those SNPs and generate something like a logistic regression model here, a linear predictor. And we typically then call the weights the betas, or in this case, the alphas that come out of that model. And then we take those weights forward into a third data set, and we use those to multiply the SNPs for the downstream data set, and we get a PRS. And I'll just make the quick point here that, and you'll see why in just a second, that the probability of D given G here in this last step is basically, or up here, is the xfit of some intercept plus the PRS, or put another way, the PRS is the logit of that probability minus the intercept, okay? Now let's switch to what a neural network could do for PRS. So imagine that you input SNPs on the left side. So you have maybe all your GWAS significant SNPs. You want to try to predict the outcome. So same basic idea, you want to build a risk model. You feed it through. So you've got all these hidden layers here in the middle. And can we pull out something that's like a PRS? Well, the answer is definitely yes. If you look at just that last layer here, the last layer is a bunch of hidden nodes, a linear combination of those hidden nodes, a bias parameter, which is essentially an intercept, making a model for predicted Y. So the PRS then that we could pull out of a neural network is essentially the logit of that end probability minus the bias, and that's going to essentially then be the sum of the weights times that last hidden node, set of hidden nodes, right? And that I would claim is a competitor polygenic risk score. Interpret it? No, right?
I guess this is not exactly like asking how to generate a polygenic risk score, but can this deal with different types of data inputs? Like, could you input SNPs as well as like other biological factors all as one input layer and see how genes and maybe like cholesterol and something else all kind of work together to predict a disease?
Yes. I'm gonna get to a little bit of that. No, that's a great question. That's what we want to get to, definitely. So I then thought, okay, I'm going to try this out. So we have our colorectal cancer data, which you've heard of before. There's about 130,000 subjects. When you pare it down to those that have complete data and other stuff, it ends up being 70 or 80,000 subjects. Okay, lots of cases, lots of controls. The E data that I'm going to talk about here includes some covariates, are the PCs, and then a series of risk factors and protective factors that are known to be in these directions for colorectal cancer. So, the standard things that as risk factors and protective factors for that particular disease. So I thought, well, let me just take a step back and not do the polygenic risk score, but let me think about an environmental risk score. So what if I wanted to feed all of those in? I didn't do the PCs at this point. I just fed in 13 risk and protective factors. They're listed over here on the left. It's a little small to see, but it's aspirin, smoking, etc. All the things I just mentioned. Ran a logistic regression, ran a neural net, training and testing. And you can see the AUC, and if we focus on the two testing AUCs here, logistic regression, about 59%, neural nets about 61.5%. So, maybe pulling out something that's a little more than just main effects of all these things. Maybe there's some interesting kinds of interactions or nonlinearities or something like that going on in there. So I was kind of heartened at that point. I thought maybe this is good. And then I turned to PRS. So again, this whole flow of identifying a first data resource. So I focused on a publication from a couple of years ago that identified 204 genome-wide significant SNPs for colorectal cancer. That was my discovery. I then used the Fiji sample, split it into training and testing. So these are the sample sizes here of the numbers of cases and controls in the training and the testing data. And I then fit this model Y on Y as disease on the 204 SNPs to generate basically a PRS. Aria helped out getting the data together, John Morrison as well. She ran the logistic regressions, and then I let Ming Ji, who was much better at this than me, run the neural networks. And then we basically looked at predicted versus colorectal cancer to see how well these models worked. And the answer is it actually didn't work very well. And we've run several versions of this. So I'm picking one out where everything I show you is going to be kind of internally consistent, same data, et cetera. Sometimes neural networks does slightly better, sometimes a little worse. This particular iteration, it was a little worse. Suggests, I think, probably there's not a lot of strong nonlinearities, et cetera, among these 204 SNPs. I then found a paper out there in breast cancer, totally different disease, but the same basic idea. They were trying to determine whether neural networks could do better for generating a PRS than other kinds of approaches. So they looked at decision trees and naive Bayes and random forests, et cetera, ran a whole bunch of models. They did different thresholds of identifying the SNPs that were fed in. So the ones I showed you, the 204 for GWAS significance, were based on having a p-value less than 5 × 10⁻⁸ in that publication. This paper, they looked at thresholds of 10⁻�3, 10⁻⁴, 10⁻⁵, et cetera. So getting different size sets of SNPs to feed in. The one that did best for them included about 5,000 SNPs that were significant at 10⁻�3. And even then, the 2 best models in terms of AUC were logistic regression, 66.5% AUC, and deep neural network ever so slightly nudging it out at 67.4%. So less than 1% better, but both did better than all of these other approaches here. They also reported the AUC for neural networks for different sizes of SNPs that were fed in. So different thresholds of significance. And for example, if they went to a more stringent significance level that only identified 2,100 SNPs that were significant, the AUC for that dropped to less than 66%. So, you know, you can see that the size of the data that's being fed in really seems to have an effect on how well these things perform. So relative to what we did, which was, you know, 204 GWAS significant SNPs, what they came up with was SNPs significant at 10⁻�3, got me thinking that maybe what's going on for us is that the 204 GWAS significant SNPs are pretty darn significant, right? They were identified in a genome-wide scan. They were probably identified because they have a pretty strong direct effect on the trait, right? They're a very selected subset. They don't need to interact with anything to get there, right? So we're feeding in a set of SNPs that maybe a priori has very low chance of having anything else you're going to pull out, allowing for.
The interactions are sort of die off as variability.
There's that too. I mean, the other thing. I mean, I'm glad you brought that up. You know, these SNPs are fairly weak effects, right? The R-squared or the heritability explained by any one GWAS SNP is fairly low, and when you look at interactions, it's even lower.
We've talked, just to clarify, the 5,000 SNPs here, there was pruning as well, right? So these are hits and then throwing out anything that's in L.
Yeah, that's a good question.
They're all independent SNPs, so this is solely finding sort of interactions of SNPs or epistasis, not sort of weights that might capture LD within a region in a signal.
I think that's true. I would have to go back and look for sure at this paper, what, you know, how much pruning they did, but yeah, absolutely. Okay. Yeah. Okay, so then I thought, well, let's throw the SNPs and the environmental factors in, have them duke it out, see what happens. It didn't do much. We're getting worse. Not good. Okay. So I was a little discouraged at this point. Again, this is one run. We've had different data sets, different adjustments. So there's still some issues. There's a lot of issues in how you pre-prepare these data and what covariates you include. And, you know, so I think, you know, basic epi and biostat principles of, you know, make sure you know what you're analyzing, what your questions are, I think is really important to keep in mind as you do this, because you can get different answers, you know, depending on how you make some of these decisions. I'm going to do a very quick sidetrack here. I talked about this a few weeks back, and some of you have heard this many times now, but we've been thinking on a whole separate thing about polygenic risk score by E interactions. And coming to the conclusion that throwing all SNPs that are GWAS significant or a larger collection and make one PRS may not be the most effective way to identify interactions, because you might be watering down true signals that are happening within a subset that truly are involved in interactions. So we thought about breaking it into subsets. How do we do that? Well, we worked with YU over here, and his student Brian Queme, who used their tool AnnoQ and Panther to identify from the 204 SNPs, those that fall into specific pathways and are overrepresented in those pathways. So 4 pathways emerged: TGF-beta, Alzheimer's, gonadotropin, and cadherin. More SNPs among the 204 within those pathways than you would expect by chance alone. So maybe something going on in terms of their collective role in how they affect downstream colorectal cancer. At this point, nothing to do with interactions, but we took those forward. Here's the lists of the SNPs. We took those forward and said, well, rather than form an overall PRS, let's generate what we call a pathway PRS. That is the sum of the effects for just the subset of SNPs that might fall in a particular pathway. Right, so there's like 14 SNPs in the TGF-beta pathway. Of the 204 we could put into a PRS, we just pulled out the weights for those 14 and created a pathway PRS. We did the same thing for the other pathways. And then came back and asked, well, do any of these pathway PRSs or the overall PRS interact with NSAIDs or aspirin, right? And if you look at the overall PRS based on all SNPs, we don't see evidence for an interaction. Odds ratio is only 0.98, not significant. However, if we focused on 2 of the pathway PRSs, we see something that looks somewhat interesting, statistically significant, and indicating that those at high risk based on just the SNPs that are in those pathways may have a stronger benefit of taking NSAIDs. That's what the negative kind of odds ratio here would indicate. Remember, NSAIDs is protective. So this is suggesting even more protection for those people that are at high genetic risk within SNPs that are in these pathways. It gets even more complicated though, because turns out there's a lot of overlap between the SNPs that are in the TGF-beta pathway based on the annotation data and the gonadotropin releasing pathway. So there's some SNPs that are unique to TGF-beta. Those are the blue ones. There's some SNPs that are unique to gonadotropin. Those are the orange ones. And then there's some in the middle that are actually both, they're annotated to both pathways. That's just the state of the knowledge up there. So we fit a few different models, but I'll focus on these ones down here. So if we throw all these SNPs together, there's 21 SNPs represented in that Venn diagram. And we see an even more stronger interaction effect. If we break it out into just the blue SNPs or the black SNPs or the orange SNPs, we see some effects, and they, depending on what subset you choose, you can see that things differ. That's not really what I want to focus on. I just want to say that's something different. Now I want to take that idea back into the neural network. So what we did this time is, rather than focusing on all 204 SNPs, we pre-analyzed them and created these pathway PRS scores. So rather than feeding in 204 SNPs, we're going to feed in just some pathway PRS scores and the exposures. So only about 20 variables going in.
So this is kind of like a form of feature engineering.
It's a feature engineering, right? So we're trying to bring in a little bit of annotation and biological knowledge to maybe pre-direct the search a little bit, right? So we have what we call the 3P PRS, which is lumping the Venn diagram together into one for the TGF-beta. There's a similar picture for the other 2 pathways. There's a lot of overlap. And then there's all the SNPs that don't fall into a pathway. That's the other. So there's 3 pathway PRSs, or if we do the more refined, the ones that are unique or overlapping, we get up to 7. Anyway, we run that through and we get something that looks a little more interesting now. So neural networks seem to be picking up maybe these kinds of interactions that we've seen. The interactions we found, way before this, just by doing some targeted analysis. But when we feed it into the neural network, it may be picking up some of these interplays now between feature engineered sets of SNPs and the environmental variables. Still, it's not earth shaking. We're not increasing the AUC by 10%. It's like 1.5%, but maybe every little bit counts. then I ran across this kind of interesting paper, and I'm almost done, so this will go quick. They basically said, can we integrate pathway knowledge into deep neural networks? So exactly what I was thinking. So here's the standard feed forward network that I've been showing you, all the SNPs going in here on the left. So nothing different there. In this one, they say, well, let's insert in this first layer, let's call that a pathway hidden layer. And for this first node up here, call that pathway number 1. We're only going to allow non-zero connections of SNPs to that node if they are actually in that pathway. So everything's still interconnected, but you force these dotted lines to have a 0 weight, and you don't update them if they are not in that particular pathway. So maybe the diagram down here in the lower right, you could think of where you've got these nodes in here that are kind of private to particular SNPs. So in our situation, it basically breaks down to this kind of a thing where you would input all your SNPs, you have these pathways defined, you have your exposures, and then you're going to run from there, and you could have more hidden nodes. This would be kind of the feature engineering idea, but rather than defining those features in advance and fixing their values, you're going to let those features be part of the architecture. And then there are some weights in here that could be actually modified as you are fitting the neural network. We haven't run that one yet. But it gets to this idea of, how to bring knowledge into neural networks and try to improve things. So there's a very well-developed area called physics-informed neural networks. And I'll thank Rima Habra, who is in the environmental health division, for a couple slides here. She's used this for air pollution. And the basic idea here is, if you want to estimate what somebody's exposed to, say, out in the inland valleys of Los Angeles or anywhere in the Los Angeles basin, we know that there's a lot of source pollution generated throughout the basin. Things happen as that air mass moves, through the atmosphere. There's chemical reactions that are happening that depend on temperature, wind speed, elevation, et cetera. And there's some well-known kinds of relationships that are happening with NOx and ozone and particulates as this air mass is moving. That generates some nice differential equations for how NOx and NO2, et cetera, might be related to each other. And the idea is you can modify, one approach is you can modify the loss function to not just be a function of the standard mean squared error, but you can work in these partial derivatives that impose some kind of a physical constraint. So now you have this dual loss function. Every time you run through, you compute the loss function and you impose upon it these kinds of additional features. And she has done some work with her team showing that they get much better predictions than a whole bunch of other ways if they use a physics-informed neural network, bringing that kind of science in, than if they do these other things or a standard uninformed neural network, which is what I've been talking about. So unpublished work, but really cool. There's a whole bunch of cool stuff out there. If anybody's interested in this physics-informed neural network, there's a guy named Steve Brunton at University of Washington that has a nice series on YouTube where it kind of takes you through. But it got me thinking a lot. And I think there are opportunities for us to think about infusing biological knowledge in the way we choose the data to analyze, the pre-processing or the feature engineering, how we set up the neural network architecture, we add nodes in there. Maybe the loss function, if we can think about augmenting the standard loss function, and the way the optimization procedure works, there may be ways to build constraints in there as well that could ultimately improve efficiency. So I'm going to finish there. I think I've come to the conclusion that we may be able to do better than standard regression for PRS. But we probably need to go down and get beyond just the GWAS significant SNPs, and the gains are probably likely to be somewhat small for a complex disease like cancer. I definitely think there's some promise for bringing in other factors and looking at things like interactions. But here, I think leveraging biological knowledge might be a key to making these things work as well as possible. So I'm really intrigued by this last bullet I have here about the flexibility of these models to allow us to think about where to infuse additional knowledge. And I think the framework seems it's complicated, but it's not that complicated. It's relatively easy to run these. I think there's lots of tools out there now to be able to think about how to do things like modify the loss function or change the architecture or things like that. So I'm really intrigued by these kinds of possibilities. I will, yeah, David.
So it's intriguing, but what about the interpretation? So, I mean, the whole idea of a polygen risk score by E is to identify populations that, you know, potentially for interventions, things like that. And if you throw it all in a model and don't understand.
Yeah.
You know, there may be some room for, again, targeted learning or other, you know, hypothesis focused type of ways of interpreting these models, but.
Yeah,
It's not the conventional additive versus multiplicative public health interactions we think about it.
No, I totally agree. I think there are some times, especially in genomics research, where prediction is what you want, right? You want to be able to build the best thing and you're going to use it downstream. So you don't necessarily care about what the inferences for how you got to the best thing. You just want the best thing and then you want to try and use that in some other way. So I think it could be good there. I don't know enough about what Kelly was talking about trying to reverse engineer the weights and try to use that in some way. But I think it's going to take something like Juan Pablo's targeted learning to be able to put a more rigorous inferential framework on it. So it's not for every problem. There's no question.
That's much document. Do you know if there's a way to do some sort of feature selection? Again, if you have the example of the PRS, it becomes important to know which are the SNPs that we should sequence at a population level to build the PRS, right? So is there a way to do that back tracing, not just figure out which are the variables that are being gathered higher, but there are efforts to features that are more important than others?
I'm not sure. Kelly was going to answer that question.
Yeah.
I haven't heard of anything of this